In this video, we will discuss about this calreticulum silent cells impaired EGF-induced invasion and migration. So previously we have seen that this calreticulum affect this EMT transition, and we have seen this from the difference in the Bradfield images between the scrambled cell and the calreticulum silent cells, and also we have seen the effect on the signaling pathways that are upregulated by EMT. So in the scrambled cells, those EGF induced cells are so in the scrambled cells which are induced by EGF shows upregulation of those EMT pathways, whereas calreticulum silent cells does not show. So now they did one more study to further confirm this effect on this EMT transition by this calreticulum silence by this calreticulum. So they checked the invasion and migration assays. So first one is about this KPN2 cells invasion assays, where we can see that for this EGF induced cells, invasion is significantly higher compared to the control, but for this CRT silent cells, the difference is not that much. And these things are quantified on the right hand side, where we can see that compared to control, the increase in the invasion is more in this EGF induced cells, whereas for this calreticulum silent cells, for both the catenaries, the increase is not that much or is not statistically significant. And when they check for the migration assays, in this case they also see the similar results. That is, for the case of the scramble cells, the increase is statistically significant, but for the case of calreticulum silenced for both the catenary 1 and catenary 2, the increase is not statistically significant. And when they check it for the ASPC1 cells, here also the results are similar, and it is also more clearly visible from these eyes also. The increase from the scramble cells is clearly visible, and this also shows in the this also reflects in this quantitative analysis. And for the calreticulum silent cells, for both the catenaries, the increase is not statistically significant. And similar results we have seen for this migration assays of the ASPC1. The results for the BxPC3 cell lines is not shown here, it is shown in the supplemental figures, but they have also said that the BxPC3 cell lines also follows the same trend, that is, invasion and migrations are silenced in the calreticulum silenced BxPC3 cells when they are induced with EGF. So this, along with the previous results, indicates that this calreticulum plays a role towards this EGF-induced EMT transition, and in the absence of calreticulum, those EMT transitions are getting impaired, and that results in the subsequent effect of this EMT-related functions such as invasion and migrations, and also the pathways that are upregulated in the EMT transitions are also impaired, and the phenotypes is also seen that is more close to the epithelial than to the mesenchymal state. So this is what they finally concluded, not finally concluded, but concluded at the end of this figure, that calreticulum plays a role towards this EMT transitions of these pancreatic cancer cells. And if you need to understand more about their experiments in detail, you can go to the reference that has been provided in the description. And if you want to see this BxPC3 results also, that too not just for the invasion and migrations, but also for the signaling pathways, and also for this Bradfield images, you can refer to the supplemental figures. I think Bradfield images figures are all for BxPC3 cell lines is there in the main figure. So for the signaling pathways and for this invasion and migration assays, you can refer to the supplemental figures. These assays could be done on the transfell. In the case of invasion, there is a coating of ECM should be there. Usually it should be the matrigel, and in the case of migration, the transfell assay is done without any coating. and the top chamber is induced with EGF. So this is how the experiment is done. If you need to understand more about this experiment in detail, you can go to the reference that has been provided in the description. So that's all for this video. Thank you for your time.